thank you for inviting me to this great workshop. It's really an honor to be here. So today I'm going to talk about some, motivated by some recent experiments that we did in bacteriophage, some recent models that we have to develop to better understand how viral expansions actually work. Probably you're all familiar with range expansion. So this is the process by which populations invade virgin territory. And this is known not to be, it's been very well studied in different species. Specifically, myself, I studied it quite well in bacterial expansions. So this is what I'm going to show you here. It's a movie of the front of a colony, of an E. coli colony expanding on an agar plate. And this is a movie, you see, it's going. What you find is that if you actually track all the lineages of the bacteria over time, so let's say 3 hours after the expansion, you look at all bacteria that are at the front, and then you trace back where they came from 3 hours before, what you find is that they all came from very few cells that were at the very, very front of the expansion. And you can actually quantify this. And what you find is that the probability, this is what I'm plotting here, the probability that an individual will actually be able to leave an offspring in the front later on is pretty much 0 as soon as the bacterium is 1 cell behind the front. So unless they're really at the front, they won't be able to make it. So this process, has been, as I said, studied in different kind of populations, but in general is called gene surfing. It's this process by which individuals that are at the front have a higher chance to stay at the front, but also to leave their offspring in the front. So this generates a feedback by which actually genetic drift can be quite strong. So diversity in this kind of range expansions decreases very rapidly. So one of the consequences of this when, for instance, people do experimental evolution is that there is this prediction of accumulation of deleterious mutations at the front. Because in general, when you look at the populations, there are many mutations that can happen. Some are beneficial, some are deleterious, but most of them are going to be deleterious. And if only few individuals, the pioneers at the front, are actually competing for the front, it's very likely that they accumulate deleterious mutation and fitness can decrease. And this has been both predicted theoretically, but also it's been shown experimentally in E. coli populations that were inoculated, let it go for like 3 days, picked at the front, replated on a fresh plate and kept going. And over time, consistently across replicates, what people found was that the size of the colony would get smaller and smaller in agreement with this accumulation of mutations that decrease actually the growth rate. So now, I guess 5 years ago, we tried to repeat the same experiment by using bacteriophage. So what I did in collaboration with Wolfram Möbius at the University of Exeter was a very, very similar experiment, but rather than growing bacteria on an agar plate, we inoculated a fresh lawn of bacteria. We put some phage, in this case specifically T7, in the center, let it grow for 10 hours, pick at the edge, re-inoculated on a fresh lawn, and kept going like this for 14 days. And what we found, this is the sequencing data, but this is not so important. What we find is more this graph here on the right, that over the course of the experiment, the phage just kept evolving to become faster and faster and faster. And indeed, by the end of the experiment, it was so fast that it actually ate the whole lawn in the 10 hours that we gave the experiment to do. So in the course of 14 days, it evolved to actually spread 5 times faster than the ancestor. So I'm not going to talk about how it does it, because actually we still don't know. It's still kind of an open question. But one thing that the result of this experiment motivated was just a rethinking about how these range expansions in viral populations occur, which is what I'm going to talk about today. So there is what I mentioned is that in this range expansions, there's this expectations that mutations, deleterious mutations will keep accumulating at the front. There is However, there are aspects of the dynamics of the wave that can limit the accumulation of deleterious mutations. And one of these is when you go from a pulled wave to a pushed wave. So maybe many of you may be familiar with this, but just to recap, a pulled wave is what you would get, for instance, in a Fischer-Kolmogorov wave. So when the maximum growth rate is at the very, very front of the expansion, which is also what we saw in the bacterial colonies that I've shown at the very beginning. In that case, it's the individuals, the closer you are at the front, the faster you will grow because you have less competition for resources and space. A different dynamic is generated when you have push waves. So push waves are a bit different, and often they're generated when there are cooperative effects in the population, so density-dependent growth or dispersal, so that actually the maximum growth rate is not at the very front of the wave, but is actually a bit behind. With the idea is that you need enough individuals around you to actually capitalize on everybody and grow faster. So push waves are known, well, to spread faster than pulled waves, but from a point of view of population genetics, they have quite interesting consequences when it comes to diversity and fixation probability. So if you actually, this is work that was done by Gabriel Birzon and Kirill Korolev now a few years ago. So if you actually label all the individuals in the wave as a function of their distance from the front, and you look at the probability that one of these individuals at a certain, labeled with a certain position, will actually eventually fix in the front, what you find is that pulled and push waves have qualitatively different behavior. So in the pulled wave, the closer you are at the front, the more likely you are going to fix eventually and take over the front, kind of what we saw in the bacterial populations that I showed at the beginning. In push waves, that's not the case because there's this competition between actually being at the very front, and so being more, having a better position to actually take over the front, but on the other hand, you're also not growing as fast, so you need to be a bit in the back. So the competition between those two effects gives actually a probability distribution that has this kind of bell shape here, where actually the individuals that are most likely to fix at the front are not really at the front, they're a bit in the back, because they have this advantage of going faster. One of the consequences of this is that actually the effective population size, so the number of individuals that actually do contribute to the next generation over time, is much, much bigger in push waves, even if the carrying capacity of these waves is the same. And this is what is briefly shown here for as a function of, in this case, dispersal cooperativity, but the same would be for growth cooperativity. So exactly the same dynamics follows whether you have a density-dependent growth rate or a density-dependent dispersal rate. So nothing, in the end, nothing really changes. You still get this higher effective population size, the more cooperative the population is. Obviously, the consequences of this, well, if you have a higher effective population size, well, the first consequence is that the genetic diversity will actually go down much more slowly. So if you have a certain number of different individuals at the front, before one of them will fix, you'll have to wait much longer. And the other consequence is that also selection is more efficient because you have more individuals competing with each other. So if you have a beneficial mutations, it's actually more likely to fix. And if you have a deleterious mutation, it's more likely to get purged by the population. So adaptation should be actually faster, which is one of the reasons why you expect expansion load to be mitigated in this kind of pushed waves. Okay, so we had this question, and the question, so we knew that this happened in this pulled versus push dynamics, and one question that came up was whether the viruses that we were actually doing the experiments on were cooperating, and we just didn't know. So we started thinking a bit about what happens when you have this phage expansion, and just to give a bit of like biological feedback on the phage that we use. This is a forcibly lytic phage, so it doesn't go through any lysogenic cycle. It just infects the host, stays inside the host for a certain amount of time, and then eventually the host bursts, creating many new viral particles that then go on and diffuse and find a new host. Also in the setup that we had, the bacteria are not motile, they're stuck where they are, and only the phage can actually diffuse. All right, so if you put this in a simple model, what you find is that we have B is the phage, B is the bacteria. When they meet, there's a certain probability that the phage will actually infect the bacteria. We call this absorption rate. And then after a certain time, tau, called the lysis time, the infected bacteria will burst, generating beta new phages that then go on and diffuse. So if you put everything together in a system of partial differential equations, you can model the dynamics of the bacteria. So in our case, we're assuming that the bacteria are already kind of a carrying capacity. So the only thing that happens for them is getting infected. Those bacteria that get infected become, well, infected bacteria. This is I. And then after a certain amount tau, they lyse. And with regards to the phage, we have that the density dependent, the dynamic of the phage is that it's just dictated by diffusion, the absorption to uninfected bacteria B, the absorption to possibly already infected bacteria, described by this parameter alpha star. And the reason why we have it is that depending on the phage, some phages do absorb to previously infected bacteria, some phages don't. So it depends a bit on the biological system that you have. In our case, they do. So alpha star is equal to alpha. And then here you have the burst of cells that were infected tau times before. So this is a time delay system of differential equations. In particular, what I want to point out here is that the diffusion coefficient that we use is actually, we don't assume it to be constant. And this becomes obvious if you look at this picture. So this is a section of the bacterial lawn that we actually use in our experiments. And what you see here is it's upside down, unfortunately. So the agar is up here. All these gray blobs that you see are cells, sectional cells. And the black dots are actually the phages that are diffusing through the front. So what you can see is that this is a very crowded environment. The phages are completely stuck in this lawn on one side. On the other side, you also have to remember that, so this is how a phage a range expansion will look like. So here, what you see here in gray is the actual bacterial lawn, and here with a razor blade, we have inoculated some phage. So over time, as the phage expands, it clears the bacterial lawn. So it's removing all this stuff that it has around it. So you can imagine that depending on on where they are in this range expansion, the phage either experiences a very, very crowded environment if they're really at the front, or actually a not so crowded environment if they're in the back, because all the bacteria have been lysed in the back. So this is why this, yeah.
Just to make sure I understand, so what are the different scales for, so do you start, maybe I missed the information, sorry. Do you start from a plate full of bacteria and then you spread like phages in it, or do both populations grow at the same time?
So they both grow, but the bacteria don't grow very much. They grow only for the first couple of hours. So by the time we are actually looking like a steady state, basically the bacteria are not growing anymore. Yeah, so you can assume that only the phage is really growing by then.
Okay. Thank you.
Yep. Okay. So if you look at this range expansion, basically you cut it vertically, you can see that at the front there will be a lot of bacteria, in the back there will be almost no bacteria. And this is why this diffusion coefficient has to be, well, we have to be careful in how to treat it. So that's what my first PhD student, Michael Hunter, did, was actually trying to understand exactly how this diffusion coefficient changes. So what he did was basically creating a plate here with a lot of bacteria in the background, which are here represented in yellow. So these bacteria, importantly, are resistant to phage. So the phage cannot adsorb to them, but they represent physical obstacles. So they would create an environment a bit similar to what I showed here, where we can actually tune the density, so change how much crowded this is. Then on top of this environment of bacteria inoculated several droplets of phage and of another bacteria that is actually susceptible to the phage. So if you then zoom, for instance, in a pair of these droplets, so here you have the phage droplet, here you have the bacteria droplet, you can actually, well, measure obviously the distance between the droplets and monitor how long it takes before you actually start seeing a clearing in the bacterial droplet. And this is a first passage time problem. So if this is diffusive, you expect a linear relationship between the distance squared between the droplet and the time it takes for the phage to get to that droplet. So that allows us to actually measure directly the diffusion coefficient of the phage as a function of this background bacterial density, which just represents these physical obstacles basically that the phage has to undergo. So if you do this measurement as a function of this bacterial density, you get this nice relationship between the diffusion coefficient of the phage D and the bacterial density B, which actually is well recapitulates what you would get in a colloidal suspension of cells. So it's a quite well-studied soft matter problem. Okay, now that we have this expression of D as a function of time, we can put it back in our expression and just see what it does to actually this range expansion. So in particular, we looked at 4 different model variants because we were really interested in understanding how this diffusion coefficient would change the dynamic of the wave. So on one side, so this would be the top row, we just assume that this diffusion coefficient didn't vary as a function of bacterial density. So that's just the vanilla case, and this diffusion rate is constant. On the second row here, instead, we do basically put in the equation that Michael found experimentally. So you find that the diffusion of the phage is low at the front of the expansion because there's a lot of bacteria, and it's high instead in the back because there's not as much bacteria over there. Then the 2 columns here, plus and minus, this go back to this alpha star parameter. So is whether in the first column is in the case in which phage can infect previously infected bacteria, instead minus is when they cannot. And that we'll see actually makes a difference. Okay, in general, if you put even just with a constant density-dependent diffusion, the kind of waves that you get, the solution of this partial differential equation system is what you see here. So you get an expanding wave of bacteria, and then 2 receding waves, sorry, an expanding wave of viruses and 2 receding waves of bacteria. Here there's uninfected bacteria, and then in the center you get this region of infected bacteria that follows the bacteria. And all this proceeds at the same velocity. All right, so the question was, is there a regime here in parameter space in which we actually do get pushed waves? And the nice bit about push waves, which is it's actually that we can determine if the wave is pulled or pushed without having to do any kind of stochastic simulations or anything, because it all depends on the actual velocity of the system, the one that you could get, for instance, just solving the system numerically, compared to the velocity of the linearized approximation of the system. So if the system is pulled, if the wave is pulled, these two are the same. If the wave is pushed, then the actual velocity of the wave is higher than what the linearized approximation will give you. So you can just look at the fraction of these two and see what you get. So that's what Michael did here. So this is a plot for a specific set of parameters that correspond to phage T7. So we have a lysis time of around 20 minutes, an absorption rate by bacterial density that is around 0.1 minutes, 0.1 phage per minute. And what he found for the different models is that indeed, so basically when the diffusion is constant, the wave is always pulled. So there's not much interesting that happens. However, when you start introducing this density-dependent dispersal and you go a very high bacterial density, then you do enter actually a pushed regime, which is what you see in these lines that enter like the dark gray region over here. Importantly, while this may seem high bacterial density, this is actually the standard bacterial density that you would use in an experiment. So when you make a LON, with an overnight culture, it would be in this regime. So it's not experimentally unreasonable. Obviously, this is for the parameters of T7, but we were interested to look in general how this would depend on the different parameters of the phage, so different sorption rate, lysis time, and so on. And what we found, so when we explored, so here there are the 4 different, it's a phase diagram basically for the 4 different models. On the x-axis, there's the bacterial density that you tune. And on the y-axis is this other parameter, which is basically the product, if you forget about Bmax, it's basically the product of absorption rate by lysis time. So it gives you an idea of how many phages are getting absorbed in a lysis time. That's more or less the quantity that you're looking at. So what we found, again, if the diffusion is uniform and actually absorption to infected cells is allowed, nothing for the whole parameter space, these waves are pulled. If you go down here, which is when you actually introduce the variable diffusion, then you find what I showed basically in the case of the T7, that at a very high bacterial density, you get in a pushed regime. What was a bit surprising is that actually you find a pushed regime also if the diffusion is actually uniform, so you're not introducing any variable diffusion rate, but you actually prevent absorption to infected cells. And this was initially, so I'm talking about this UDM minus problem here, and this was a bit surprising. But in particular, where you find this push regime is when the absorption rate by the lysis time, by the bacterial density is very, very high. So when you get a lot of phage absorbing in a lysis time. So why does this happen? Well, with hindsight, then you think back about the process and then it becomes obvious why actually it happens. So the reality is that there are actually, if you want, 2 different mechanisms that hinder diffusion in the phage, which are a bit sketched in this plot. So if the phage is just freely diffusing, well, they will just, it will have a certain diffusion coefficient over a certain amount of time. Then what I talked about, and the thing that actually Michael measured experimentally, is this explicit effect. So the fact that if you have all these bacteria around you, the phage has to navigate around these bacteria to be able to diffuse over the same amount of time. And the hindrance to diffusion is dependent on the bacterial density according to the equation that Michael basically measured experimentally. But there's actually a third effect, which is due to the fact that when the phage adsorbs to a bacteria, then it's stuck in the bacterium for a certain lysis time. And while this is not explicit in the sense that we don't have to modify our system of differential equations, it naturally emerges because you have all the phage, and some phage will just be removed for a certain amount of time by the population and not be able to diffuse. So one can do a simple mean field argument, and what you find is that the hindrance to this diffusion depends in this way from the absorption rate, lysis time, and bacterial density. Importantly, it still depends on the bacterial density, because the more bacteria you have, the more the phage will have a chance of getting stuck there and not being able to diffuse, but also depends on this alpha tau. So if I have to summarize everything, basically you start from this UDM plus model that gives this effective diffusion rate actually looks a bit like this, because this implicit effect is always there, it's unavoidable. As soon as you have an incubation time, so the phage will, or the virus, whatever, is going to get stuck there for a while, you'll have this. You will always have that the front diffuse lower than the back. If you then introduce the explicit effect, so you go to the VDM plus model, you go from this black line to this red line, because now the phage at the front not only is stuck in the bacterium, also has to navigate around the bacterium. So there's this physical effect that creates even more an imbalance between how fast the phage diffuses at the front versus the back. If we go from the UDM plus model to the UDM minus model, then what happens is that you're actually shifting this black line to the right. And the reason why this happens is that, so if you think about this blue effect, this this reduction in the absorption rate. So this is due to the bacteria that are actually able to absorb the phage, right? So in the UDM plus model, also the previously infected bacteria can absorb the phage. So you feel this reduction in diffusion coefficient already here in this light gray region, which is the region of infected bacteria. Instead, if you go to this model, the UDM minus model, now the infected bacteria cannot absorb the phage anymore. So the phage that is up here doesn't feel this effect. It's only if you're at the very, very front where the uninfected bacteria are that you feel this, which is why this is all shifted basically to the right. And now you give a chance actually to the phages that are in the back to diffuse faster and catch up with the front, which is why you get the emergence to the transition to the push wave. Then obviously if you combine both, you get the largest effect, which is this pink line over here. But basically, to summarize, this gives you an explanation of why the transition lines actually look as they look in this plot. So in the VDM plus model, we said the main effect is this explicit effect due to steric interactions, basically. So volume excluded interactions between the phage and the bacteria. So this is controlled, as you see, well, in this red equation over here, mostly by bacterial density. And indeed, you get transition lines that are vertical, so they depend only on the bacterial density. In the UDM minus model, the steric effect is not there. We didn't put it. And there's only the implicit effect, which depends on this product alpha TB, which is exactly, so a constant alpha TBs give you these lines that then give you then the transition from pull to semi-push, then eventually push waves. I don't know how I am with time because I realize it's
yeah, 5 minutes.
I have 5 minutes. Okay, so I'll just, this is work that Michael did. I'm just very quickly going to show where we're going from here. So a question showed this before, the bigger result of these push waves from a population genetics standpoint of view is that you get a much larger effective population size. So something that we were wondering was whether, so we have found that our viral expansions undergo push waves, but what's their effective population size? We don't know yet. And in principle, it's not obvious that you can map the push waves that we find to like the push waves that people normally find in single species colony one by one, because they're very different systems. First of all, well, as we said, this is an emergent dynamic from the system of coupled differential equation versus a single reaction-diffusion model that we have. There is a time delay due to the lysis time. It always complicates things. And, but most importantly, I would say is that unlike in single species range expansions with a cooperativity parameter, where this cooperativity parameter is the only knob that you can basically tune to go from pull to pushed, our cooperativity is a function of everything else. So as soon as we change alpha, tau, any of these parameters, we're also changing many, many other things in the system. So it's not clear exactly how to map one to the other. So this is what actually Nikhil, who is a second PhD student in my lab, has been working on recently, both using simulation, agent-based simulations, and also stochastic partial differential equations. In particular, he focused on this UDM minus model, which is just a simpler model where the only, theoretically, the only parameter that controls whether the wave will go from pull to push is this product alpha tau b. So what he found after doing both simulations and some analytics is that things are obviously a bit more complicated than what you think. So first of all, so if you look at this parameter k, which is this product alpha b naught tau, which should be this cooperativity parameter, as you increase it and you look at this is the ancestral distribution, so this bell shape. So this is the probability that the individual that fixed eventually came from that position time before. So this is what I was saying, that in push wave should look like a bell shape. So indeed, what you find is that if you increase k in general, this bell shifts towards the bulk of the way. So it shifts towards the left, which is good. Importantly, you can change K in two different ways. You can increase absorption rate, or you can increase lysis time, right? So this is what is shown on the left. We are increasing absorption rate going from the blue to the yellow. Instead, on the right, we are increasing lysis time going from the blue to the yellow. So they always shift to the left. But how they do it is very different, and how the profile also changes is different. So if you change absorption rate, what happens is that actually the profile gets shallower and shallower, which is the opposite of what you would expect in a push wave. So in a push wave, when you go to more push, the profile actually gets steeper. And this has a consequence, because actually when you go and measure the effective population size, then you get two opposite effects. So as I said, normally in start and push waves, you expect it to increase cooperativity and the effective population size will go up. Here we find that if we increase absorption rate, actually the effective population goes down. Instead, if you increase the lysis time, the effective population size goes up. And again, this should, in principle, they should both go from pull to push. Long story short, you can actually find a rescaling so that everything collapses in a nice way. But you really need to take into account the fact that these profiles are getting stretched around, and that the carrying capacity is also changing as a function of this parameter. So if you rescale the effective population size by basically B naught, which is the number of bacteria that you have at the front, multiplied by the burst size, so that gives you kind of a more or less a rule of thumb for their carrying capacity. And you also rescale by L, which is how much the phage can diffuse in a lysis time, so that gives you a spatial scales. So if you rescale by these things, then everything works out nicely, and you get that then all the lines collapse, and you can and you get actually a map to the standard push waves. But you need to go through this rescaling basically to do it. And then just really briefly, the other aspect that is known between pulled and push waves is how the effective population size scales with carrying capacity in the population. So importantly, when the wave is fully pushed, the effective population size is proportional to the carrying capacity. When it's semi-push, you get an exponent that is less than 1, and then when it's pulled, you get a logarithmic correction. So we checked this for our case, in particular as a function of B naught, which is the best proxy that we have for carrying capacity. And we do find the same behavior. Sorry, so this is minus alpha, but it's the same. So we do find that when the population is going to very pushed regimes, indeed, you get an exponent of -1. So the effective population size is proportional to carrying capacity, or something proportional to carrying capacity. And then if you decrease, you go to lower exponent. So this also seems to work out. With this, I'll just stop. So I hope I convinced you that the feedback between density-dependent diffusion and growth dynamics of phage and bacteria can give rise to this emergent, like pushed wave transition. This has really two physical origins. On one side, just the fact that the phage is in a very crowded environment, so it has to bypass obstacles around the way. And the other origin, instead, it's more due to the actual underlying infection dynamics. So it will be present also in a virus, for instance, infecting a tissue, like in different conditions. And then we're still working, but it seems that there is hope to actually map these pushed waves to like standard push waves that people have studied before. Thanks.
Thank you very much. You do have a question?
Yes, if no one else has it. So there's no time for evolution of bacteria, I guess. Because
in our experiment, no, yeah. So how we do it, we specifically did it. So every time we put the lawn down, we take from the stock for the lawn, because we were really interested in the viral evolution and not the co-evolution. But you can, in principle, do it so that they're both co-evolving.
Yeah, because if you start having bacteria that are more and more resistant, then it becomes more and more like difficult for the phages, and only those at the tip would make it, and so you could go back from a
we haven't
push
like
to pull again.
It could be, yes. So what often you do see in this experiment is that you do get colonies that inside the plaque that are clearly resistant because they're growing, but then often you see them getting wiped out. So the phage is already in over 10 hours basically evolving to wiping out the rest. So it's an arms race every time. So it's a bit unclear what could happen, but it's something that we would like to eventually do once we understand what's happening in the simple case.
Thank you.
You may have said it and I missed it, but I was wondering how do you define the effective population size in the models? Is it in terms of coalescence or?
Oh yes, sorry, no, I didn't mention it. So we do it just by looking at the. So we inoculate. We start basically with a population of like 50-50. And then we just look at how the heterozygosity is decaying as a function of time, and then take basically the exponent of that as a definition of effective population size. This is in the simulations. In the actual stochastic differential equation, it's somewhat similar. You still define basically the heterozygosity in your system and see how it decays over time.
Thank you. So I suggest that further discussion are postponed to the afternoon. And thank you very much again.
Thank you.
Very nice talk.